Last episode we pulled over 1,000 real EGFR inhibitors. But being active isn't enough, a molecule also has to behave like a drug, absorbable, not too greasy, and free of troublemaker substructures. Today we run 2 classic filters over that whole set and watch the pile shrink to the compounds actually worth pursuing. Filters are cheap, lab assays are expensive. The first filter is the most famous rule of thumb in the field, Lipinski's Rule-of-Five. It says a molecule that's easy to absorb orally usually has a molecular weight at or under 500, a log P at or under 5, at most 5 hydrogen bond donors, and at most 10 acceptors. 4 numbers. 1 violation is tolerated, 2 or more, and the molecule gets flagged. First we compute those 4 numbers for every molecule. RDKit derives them straight from the structure, so one function turns each SMILES into a row of properties, and we join them onto our table. Now every one of our 1,000 plus EGFR inhibitors has its weight, greasiness, and hydrogen bond count sitting right next to its potency. Now the rule itself. We count how many of the 4 limits each molecule breaks, and keep everything with at most 1 violation. Run it, and 966 of our 1,065 compounds pass. Most known inhibitors are already drug-like, but not all, and the ones that fail are worth knowing about before you spend money on them. The second filter catches a different problem, PAINs, pan-assay interference compounds. These are substructures that light up as false hits in assay after assay. RDKit ships a filter catalog for them, so we don't write a single pattern by hand, we just ask, does this molecule match anything on the list? 131 of ours do, and out they go. Chain the 2 filters and you get a funnel. We start with 1,065 active molecules, keep the 966 that are drug-like, then drop the PAINs matches to land at 843 clean, promising compounds. We save that set, it's the cleaner pool we'll search and model in the episodes ahead. That's how a real campaign avoids wasting effort on molecules that were never going anywhere. So we turned 1,000 actives into 843 drug-like, alert-free candidates, using 2 filters and a few lines of RDKit. Next episode, we start searching, take one known inhibitor and find its lookalikes in the set, using the fingerprints and Tanimoto similarity we built back in episode 2. See you there.